rather than one semester. And I think it would be onerous to ask you to review everything that's in that whole textbook and just say, hey, the final's comprehensive, so it's all going to be on there. So the final will be comprehensive, but I'm going to try to direct you to the particular kinds of questions that are going to be asked. And so here's what I'm thinking. I encourage you to give me feedback, either now or by email or whatever, if you think this is suitable, which is that the bulk of the questions will come from the previous midterms, the practice midterms, a practice final, which we will distribute to you, and the assigned problems. So those will be familiar questions to you. You will have already seen them. You will have already been studying them. And particularly with regard to questions that have come up on previous midterms, you should have already given a lot of thought as far as what the suitable answers for those questions will be. So those will be maybe half of the questions, maybe a majority of the questions. We will be including some content from the material after the third midterm. Obviously, that's not a huge amount of material, but it will probably correspond to about 10 to 15% of the final exam. Any commentary about that rough plan of action? I would begin to talk to you about particular subject matters that will be the focus, except I haven't written the final exam yet, so I'd rather wait to talk to you about that until I have a much more concrete idea about what's going to be on it. Yeah? Yes. Chapter problems, smart work problems. Right. Other comments? All right. Commentary about midterm 3. Okay. So let's move on to our discussion about control of gene expression. And last time we were talking about understanding the role of repressors and activators and knowing the different ways that effector ligands can control gene expression. And then we got into the issue of what an operon is and how the lac operon and the trip operon work. We're going to get back into a little bit more detail about the lac operon. A lot of you weren't here last lecture, so we'll just kind of get into some of the particulars of the lac operon and its engagement with the DNA sequencing upstream of the operon. Okay, so this should be familiar to you by now. These are the different mechanisms that the cell uses to alter protein concentration. Your textbook has a kind of a different take on this, but it's basically the same concepts. And what they call this is gene regulatory processes. But these are essentially the same types of alterations at the molecular level that give rise to changes in protein concentration. And so you should be familiar with all of these different steps. And it begins, obviously, with the expression of particular genes and whether or not DNA is going to be accessible to RNA polymerases for the generation of an initial mRNA message, and the rate at which that RNA message, that initial transcript, is processed into a mature mRNA. These mRNAs are subject to control by alteration of the rate at which those are degraded. And then those mRNAs influence protein concentration because of the fact that they are read out by the ribosome and translated into protein sequences. This is not the end of the line with respect to control of protein concentration, however, because once a protein is made, we can control the rate at which that's degraded, and we talked about the role of the ubiquitin system in altering that. And when we talk about concentrations of proteins, I think what we really oftentimes mean is the concentration of active protein species. And that is altered by post-translational modifications, which obviously, again, play a role in protein degradation. But this issue of protein modifications gets into another form of control that this image in the textbook doesn't mention. So what's kind of missing here that we talked about before? So post-translational modifications affect not only the extent to which a protein is degraded, but what else can they do to control levels of the activity of proteins in the cell? Yes?
Regulate transcription?
Not so much transcription, that's more of an upstream kind of effect. Post-translational modifications of transcription factors can influence that, but I'm talking about a more general issue that relates to a lot of different proteins. Yeah. I'm sorry? Ubiquitination is a post-translational modification that directly influences this pathway right here, whether or not the protein is going to stick around or whether the protein is going to be degraded. Yes, sir?
Can affect the amino acid residues in the active site of the protein?
For sure, yes. And that also is missing, that the protein modifications can influence the extent to which a protein is active or inactive, and we talked about particular alterations that influence enzyme activity. So that's critical. Anything else? What about glycosylation? What is that often used to control? Yes. Right. Localization. So that's kind of the missing element here, that these post-translational modifications can direct proteins to subcellular organelles or for export. All right. So we talked a lot about regulatory proteins that influence transcription levels. So we're going to be talking about this issue right in here, proteins that alter the extent to which RNA is being created. And so we talked about repressors and activators and the DNA sequences at which these proteins engage the DNA. Your textbook mentions cis-acting factors versus trans-acting factors. And I have to say that from the standpoint of chemistry and as a bioorganic chemist, we don't often use these terms. I'm guessing a lot of you are in molecular cell bio, and I'm wondering whether or not these terms come up often in that class. No? Cis-acting factors and trans-acting factors? It's not necessarily something that I'm all that comfortable with, but I do see it in the biology literature quite a bit. And when we talk about trans-acting factors, we're usually referring to a molecule such as a protein, such as a transcription factor, that can move throughout the cell and influence gene expression. Whereas a cis-acting factor is a biomolecular entity that is in close proximity to the gene that's being regulated. And generally what we mean by these cis-acting factors are the regulatory DNA elements themselves that are upstream of these genes. So the binding sites on the DNA, those particular sequences, would be cis-acting factors, and these repressors and activators would be trans-acting factors. So this figure here gives some idea about what those terms relate to. A transcription factor would be able to diffuse and then bind to DNA. These are trans-acting factors, whereas that regulatory DNA sequence itself, that's referred to as a cis-acting site. Okay. So let's get back into how the lac operon works. And I'll encourage you to go back and study some of the previous figures that get into some other aspects of the lac operon. But I'll just remind you that the lac operon controls the expression of a bunch of genes that are capable of metabolizing lactose. And so the cell, when glucose is present, typically doesn't rely upon it. But when glucose levels are low and lactose is available under those two conditions, then the cell will say, okay, let's use lactose as a carbon source, and so it needs to turn on the genes that are capable of doing that. So here's the promoter element that's upstream of the operator that controls the expression of those set of genes. So in order to express those genes in that operon, RNA polymerase needs to sit down on that promoter. We talked before about a repressor protein, the lac repressor, that can respond to the presence of lactose and will sit on that operator and prevent RNA polymerase from associating and generating the mRNA message. Also in proximity to that promoter is another regulatory site on the DNA that is a site for binding by cyclic AMP receptor protein, referred to here as CRP. So this is capable of sensing the presence or absence of glucose. Because when glucose concentrations are low, cyclic AMP levels are high, and CRP, when it's associated with cyclic AMP, is capable of binding at the CRP binding site. So let's see the 4 different conditions with respect to presence of lactose and glucose and its influence on transcription. When lactose is absent, the lac repressor is capable of binding. I'll get into some of the details again in just a bit. When that lac repressor is there, transcription is off. When lactose shows up, but glucose is also present, we have a condition in which the lac repressor will bind to lac and be dissociated from the operator sequence. CRP will not bind here because cyclic AMP levels are low. So RNA polymerase is capable of binding, but at a pretty low level, and so only a very small amount of transcription is evident under these cases. It's really when lactose is present, glucose is absent, that we get the perfect conditions for expression of this operon. RNA polymerase binds, it's actually recruited to the site by the presence of CRP, and now we get extensive expression of the lac operon gene. So this is one of the ways in which multiple signals are integrated in the cell. We want to be responsive to the presence of lactose, certainly, but only under some conditions when glucose is absent. Okay, so that binding of the lac repressor has its influence on the ability of the RNA polymerase to move through by physically altering the architecture of the DNA, of the chromatin in this region. And it does so by dimerizing, binding to 2 sites, complexing with itself and with DNA, and in so doing, closing a loop within the DNA. Here are 2 sites for lac repressor binding, right here and right here. And the lac repressor is capable of binding at either of those, but when one lac repressor dimer is bound at one site, it's capable of complexing at that second site, and when it does so, it induces this bending and curvature in the DNA, which influences DNA polymerase activity. And we have these beautiful detailed structures of the influence of the lac repressor on the DNA architecture itself. And you can see here that we're not looking at typical linear B-form DNA. In the presence of this lac repressor, we're actually taking a look at a highly bent form. And so this is the crux of that bending that's allowing that DNA to be looped out when all the copies of the lac repressor are there. So here we see that in the presence of a suitable ligand, lac repressor is capable of binding to DNA. So where in the DNA do you think the lac repressor is binding? And what is it recognizing? Any ideas? Probably not just associating with the overall architecture of B-form DNA, or even DNA in a curved form. How do you think it's engaging with DNA? To what extent do you think it's specific? If it's specific, where is the origin of that specificity? Any idea? Yeah.
Probably near the promoter regions. I imagine it wants to bind all the other proteins binding there.
Okay, right. So it needs to be specific to the particular promoter upstream of the lac operon and not all the other promoters that are going to be present in the genome. So there is a particular sequence that it needs to read out. And we talked a little bit last time about these elements of the protein structure that are capable of sequence-specific recognition of DNA. So if it's going to be sequence-specific, what's the nature of that interaction? What needs to happen? Yeah. It has to look at the bases. And this is a theme that we've talked about quite a lot. Bases are not necessarily so accessible in B-form DNA. So let's pursue this a little bit. What part of the DNA do you think this protein is engaged with? We talked about general structural features in B-form DNA. Yes?
Could it be the major groove?
Yes, exactly. The major groove. Was that just a wild guess, or is there a particular reason why you suggested it?
Well, it can then go into the bases and form hydrogen bonds being paired.
Great. So the protein actually tickles the DNA. It reaches into the crevice to pull out the information that it needs. How does it know what sequence is there? It's got to reach in, and it's looking for a particular pattern of distinctive chemical functional groups, often in the form of a hydrogen bond arrangement. And so here it is. And then, why does it bind in the major groove as opposed to the minor groove? Well, here are the presentation in the major groove. Here's the presentation in the minor groove. You can see this groove is wider, this groove is narrower, right? Major and minor. And here are AT sequences and GC sequences. So the bases are available in either the major groove or the minor groove. You can tell that bases are there. But can you tell which bases are there? Effectively, only in the major groove. And if we label atoms that are being exposed, and label them as either being hydrogen bond acceptors in green, hydrogen bond donors in pink, or hydrophobic entities, you can see that this right here, an AT, looks very different than a TA. Right? That pattern is distinct, going from left to right for AT versus TA. And obviously it's distinct from a GC base pair. But we need to be able to discriminate between an AT versus TA. And we can do that in the major groove. Look over here in the minor groove. Yeah, we can access those bases, but there's really no chemical information that allows us to get good preference for one of these versus the other. They both present a couple of hydrogen bond acceptors, and so there's no good way to discriminate between these. And it's the same thing with GC. We can discriminate it effectively in the major groove, not so much in the minor groove, which is why very often when you see transcription factors bind, they need to amp up their binding affinity, they need to amp up their binding specificity, they improve their affinity by binding in a dimeric or tetrameric form, and they improve their specificity by going after the DNA and reaching into the major groove. Yes?
When one dimer binds to a part of the DNA, does the second dimer bind at the same time, or is it one after the other?
It's probably sequential, but probably very closely associated. And that's kind of the feeling that you get here from this cartoon. Whether or not this cartoon was drawn with a very careful attention to whether this is sequential, I can't be sure, but this seems reasonable to me, that in this case, a dimer of dimers, first one dimer binds at the site, the second dimer is floating around, and then as the DNA is being bent, it ends up hitting this other operator site. Yeah.
Is it in any way a cooperative process? Because the reverse arrows are like a little different, which might imply.
Yeah, I would imagine that it's cooperative, and I'm guessing that there is beautiful biophysical literature that talks about investigating the nature of that cooperativity. I'm not necessarily familiar with that literature. Yes, sir.
I read in the textbook that protein DNA binding, there's like an alpha helix involved. Is that here or is that something else?
Yeah, so I got into this a little bit in the last lecture, and that's an important issue, because if we're going to reach in to the DNA and feel it, engage it, we need to have an organized protein structure that is pre-associated in its proximity to have its side chains extend into this region. And there's a number of different tricks that the cell uses to create these discrete little entities that are well folded but are not full domains. And here, nature really has to provide a great balance between having something that's substantive enough to be well folded, but not so large that it can't reach in. Some people have nice, delicate fingers, and they can manipulate things really well. I've got big, stubby fingers, and I don't have the kind of acuity and physical dexterity that my kids do. So when something needs to be manipulated, I ask them. It's kind of the same thing. We need something that's well organized, but not so massive that it can reach in. So this helix turn helix motif is something that I talked about last time, and there's a variety of other protein structures such as leucine zippers or zinc fingers that can be used to stabilize a compact piece of protein structure in the proximity of the major group. Yeah. Other questions about this? All right. So we need to make this lac repressor responsive to the presence of the saccharide lactose. And I talked last time about how this IPTG, which is a sugar analog, when it binds to the lac repressor, induces this major conformational change. So we can use this effect of the binding of IPTG to drive the disassociation of the lac repressor to control gene expression. And so I'm going to echo some comments that I made last time about using the lac operon to control the expression of recombinant genes. And last time we talked about the fact that many of you have expressed recombinant genes in a bacterial host system. You grow up the E. coli, you dump in the IPTG. What's going on? This is what's going on. So here is how the lac repressor typically works. So let's say in its native context, there is a coding sequence for one of these proteins in the operon, LAC-Z. Okay, here's the promoter, the lac repressor sits there, and then in the presence of IPTG, that induces a change in the lac repressor, boom, off the repressor goes. RNA polymerase can now associate with the promoter. Now we get our mRNA for LAC-Z, and then we get the LAC-Z protein. That's the way it normally works. We don't care about LAC-Z. We want to create some wonderful protein that we're interested in. My student Antalya wants to create this fungal chitinase, and she's not going to grow up fungus and harvest it and try to purify the enzyme out of it. No, she's just going to order the gene from one of our collaborators off in the UK, and then she's going to take that gene and then try to express it in bacteria. How do we accomplish that? We take a plasmid that's got that LAC-Z coding sequence and the control regions, and we just remove that LAC-Z gene. We don't care about it. Cut it out using restriction endonucleases, and now we put in our target gene for our chitinase or other protein of interest. We put it right downstream of that very same promoter. So it is inducible by the same kinds of conditions that can normally be used to drive expression of LAC-Z. What does that mean? We bring this into the bacterial cell, the LAC repressor is present, it sits down on the LAC promoter, and when we want the expression of that protein, we dump in our IPDG, RNA polymerase engages the promoter, and churns out our mRNA for our protein of interest, and we harvest all the bacteria, and we've got plenty of our protein. So this is how we can use the LAC operon to our benefit. So this demonstrates the fact that you can take the existing regulatory architecture and you can do surgery on it to alter the gene that's expressed. But you can do the opposite to really great effect as well. You can take a particular structural gene and you can alter the regulatory region and find out, what is going on with how that regulatory region works. So, for example, we can take a structural gene, swap it out for a reporter gene, something that we can see, a protein that we can visualize, and then just introduce this whole regulatory region and reporter gene into a biological system of interest and find out where that protein is being expressed. And we can actually use the LAC-Z gene itself. We can use that particular enzyme to promote a color change so that we can see exactly where that gene is being expressed. We take the LAC-Z gene, we put it under control of an interesting regulatory region, one we would call MYF5, and we ask the question, where are genes expressed when they're put under the control of MYF5? Here's what the experiment looks like. We visualize where LAC-Z is being expressed with this colorimetric assay. We look at the particular tissue types where we see the LAC-Z reporter gene, and we figure out that it's being expressed preferentially in muscle cells. So this is really nice. We can use these reporter genes in a variety of different ways, and we can visualize them in different ways depending upon what gene is being expressed. So for the LAC-Z gene, we can do an in situ colorimetric assay. We can feed substrates into that enzyme and visualize where that enzyme is being expressed. We can create really wonderful fluorescent probes or spectrophotometric probes. For example, luciferase is a really nice reporter gene, and we can use that enzyme to turn on fluorescence of a substrate. That's a really great one. Enzyme that provides antibiotic resistance can be used for genetic selection, but this is one that I'm pretty sure that many of you have seen these dramatic images of. We can put a reporter gene for a green fluorescent protein under control of one of these promoter regions and see where in living systems this protein is being expressed by visualizing directly where this fluorescent protein is located within a living system. And the lights are a little bit bright in here, but this shows where the green fluorescent protein is being expressed in a mouse embryo and in an adult mouse. So this looks at the presence of the GFP protein throughout all stages of development. And this indicates that you can actually get GFP to be expressed everywhere if you put it under control of a promoter for a protein that is typically expressed throughout the living system. What kind of gene would that be? Well, we call these housekeeping genes. This is just a kind of protein that would be needed in every single cell, something like actin, which is an important component of the cytoskeleton. So if we put GFP under control of the actin promoter, it's expressed everywhere throughout all stages of development. It's kind of interesting, when I was taking a look at this study, I realized that GFP actually influences the extent to which a fertilized egg cell will turn into a blastocyst. And if you overexpress GFP at high levels, it'll actually really alter the viability of those fertilized eggs, which is something that I did not know. So it doesn't necessarily always have a minor impact in a living system, but it can be used really nicely to visualize where genes are being expressed. And this is an example of that. This is also expression of a reporter gene in a mouse, a living mouse. This is using luciferase. Okay, so this is an enzyme that generates a fluorescent response in a substrate. And you can see that that luciferase reporter gene is being preferentially expressed right in the eye tissue of this mouse, but only in certain stages of development from day 1 to day 4, and by day 8, the expression of that gene is shut off. So this indicates how to use reporter genes to monitor patterns of gene expression. Okay. Any questions? All right. So now that we've kind of gotten a feeling for how the expression of genes are controlled in prokaryotic systems, let's move on to figuring out how genes can be regulated in eukaryotes. So there are really big differences in eukaryotic gene regulation, and a lot of those differences has to do with this simple statement, that access to eukaryotic promoters is limited by the structure of chromatin. So if we're talking about a pattern of genes or an operon, let's say in a bacteria like E. coli, those regulatory DNA elements are pretty much sitting out there open to all of the regulatory proteins that might want to come along and engage with them. But we talked quite a bit about the fact that there's so much DNA present in a typical eukaryotic cell that it's absolutely crucial to arrange for a compact architecture of that DNA. We need to organize it around nucleosomes, and we need to provide it into small bundles and fibers and really compact it. And as we do that, we actually prevent its ability to be bound by regulatory proteins. It's the physical architecture of the chromatin that means that those promoters aren't generally available. That means that the cell actively has to work to open up those elements, allowing things like activators to bind, allowing the RNA polymerase II to come in and generate the mRNA. Because of the compaction of eukaryotic DNA, gene expression is generally shut down. The major component that helps to organize DNA in eukaryotic chromatin is carried out by the histone proteins, and we talked quite a bit about those, how they arrange themselves into nucleosomes, and that really allows for the compaction of DNA. So we can modify these histone proteins, and we can alter the affinity of the histones for DNA and alter chromatin architecture. A big modification is carried out by histone acetyltransferases, introducing acetyl groups onto lysine side chains in those histones. And as we introduce those changes, we open up the DNA, and it allows for recruitment of other components of the transcriptional machinery. It allows remodeling of the chromatin structure, and it allows the activation of genes in that region. So if those histone acetyltransferases play a role in activation, if those are the family of enzymes that can activate, when a eukaryotic system needs to repress those same genes, how do you think that can be achieved? Yeah. Okay, so what we need to do is remove these marks, these histone acetyltransferases. So that we need to undo those chemical modifications. So in this case, it's the histone deacetylases that are the enzymes that are responsible for undoing that chemical modification. So we saw before that there can be complementary families of post-translationally modifying enzymes that can either introduce or remove chemical modification onto a protein structure. So what was the other big example that we talked about last week? Regarding two families of enzymes that either introduce or remove.
Phosphorylases.
Okay, kinases.
Kinases.
And what was the other family? So a kinase will introduce a phosphate group, and then the phosphatases. So this balance between histone acetyltransferases and histone deacetylases, introducing and removing these covalent modifications, is similar to kinases and phosphatases, introducing those changes onto protein structure. I'll mention in a bit the other major modification into DNA, which is used to control gene regulation, and that's the introduction of a 5' methyl group onto the cytosine nucleophase. And that's in a bit. So overall gene regulation in eukaryotes, we really need positive regulatory mechanisms. We need to undergo an activation event to allow a transcription. Regulatory proteins and protein complexes are larger, and that just makes sense because typically eukaryotes are responding to a lot more messages and need to integrate a lot more signals. Developmental pathways are a lot more complicated in eukaryotic systems. And then transcription and translation are physically separated events. And this is also something that we've emphasized previously. Why is that different in eukaryotes than prokaryotes? Transcription and translation are separated in eukaryotes.
In prokaryotes, they both occur in the cytoplasm, but in eukaryotes, when it happens in the nucleus, while that won't happen in the cytoplasm.
Exactly. Right. So transcription is carried out in the nucleus. In eukaryotic systems, the mRNA needs to end up in the cytoplasm in order to carry out translation. So those are two separate locations within the cell. Yes?
What type of access do we have to gene promoters in like lab type settings? So is it really difficult for us to study them because of the compacted nature?
We can introduce all kinds of variations in promoter sequences in a laboratory setting. So this is really a critical tool in molecular genetics, is being able to swap out regulatory elements or introduce specific mutations in them. And now we have this whole area of systems biology. I think there's some students who are participating in the iGEM competition, where you're like moving regulatory elements in and out from one system to another, and you can cut and paste these things in many of the same ways that you can cut and paste genes and introduce mutations into, let's say, enzyme active sites. Yeah, plenty of opportunity for manipulating them. All right. So let's take a look at the molecular mechanisms by which we're going to activate a region of the chromatin. So this is what it might look like in a highly condensed state, with the DNA wrapped around these nucleosomes. Let's remember these are composed of histone proteins. And you can see that some of these DNA regions are really sequestered. We can pull these individual nucleosomes apart from these fibers and exposed histone octamers and the DNA wrapped around the outside. And these we refer to as decondensed chromatin, and we can begin to access the DNA on those, and we can begin to think about ways to engage particular sequences on those DNA. So transcription factors, for example, just like the regions that we were talking about with the black operon, these need to bind to particular DNA sequences. So transcription factors will need to move in to these nucleosomes and bind open sites on them by pushing aside where these nucleosomes are located. So some of these are directly upstream of where the RNA polymerase would want to bind. These are these promoter regions. And some of these regulatory regions where transcription factors might want to bind may be far away, and we would call those enhancer regions. Once those transcription factors are able to push aside the nucleosome, access the DNA directly, those can be sites for assembly of protein complexes that then cause alterations in the DNA architecture and bring the RNA polymerase in to begin expression of those downstream genes. So how do we do that? How do we make alterations into the nucleosomes themselves? There are 3 different ways that the textbook indicates that that can happen. One of them is, as we were saying before, through post-translational modifications, in particular, acetylation of histone lysine groups. Methylation also introduces changes into the histones, and in certain cases we see phosphorylation is also important. And these chemical changes can alter the affinity of the histones for the DNA. There's also processes by which the nucleosomes can be pushed to one side or the other, or even physically removed to open up binding sites for transcription factors. And then the textbook also mentions that a typical histone can be replaced by a variant protein structure with altered interactions to DNA. I consider this process to be quite mysterious. It's not something that I've had any familiarity with before, so I'm quite curious about it, and there really aren't any details that are available in the textbook. So much familiar with this. This is a new one for me. Okay, so let's really get at the issue of where our previous discussion in post-translational modification meets the topic of control of gene expression. We're going to take a lysine side chain and we're going to acetylate it. And we're going to use one of the member of these enzymes, the histone acetyltransferases, to create this modified lysine structure. And as we do that, we're going to change the charge state of that lysine side chain from positive charge state to a neutral one as we acetylate it. And that's really going to have a major influence on the affinity of those histones for the DNA that's wrapped around it. Again, those modifications can be reversed by the action of histone deacetylase enzymes. So what's going to influence whether or not a particular site on a histone protein will be a substrate for a histone acetyltransferase and whether or not these modifications will happen? Oftentimes, activator proteins will be able to access the DNA. They will recruit a complex that includes histone acetyltransferases, and these enzymes will then go to work and do surgery on the surrounding chromatin structure, making sequential modifications in the proximity until there's been a substantial change in the organization of the histones that alters the chromatin. And similarly, a repressor protein might be able to recruit another complex that includes not histone acetyltransferases, but histone deacetylases, and you can understand pretty quickly how the reverse of this reaction would then do similar modifications that would serve to constrain gene expression in that region. Both acetylation and methylation are used to alter histone proteins. Histone acetyltransferases, histone methyltransferases, both play an important role. I'll ask you to consider what are the major differences between putting on an acetyl group and a methyl group onto a lysine. So you might want to consider that carefully. And I'll ask you to think about whether or not methylating a lysine side chain is going to remove the positive charge site, the same way that acetylation might. And I'll ask you to think about whether or not you can do sequential rounds of methylation at a primary amine side chain, and whether or not you could do multiple acetylation onto a lysine side chain. So those are some of the key differences that you might want to think about in the action of these two enzymes. Following these modifications, we have new acetyl sites, new methyl sites, and so as we put in these modifications, these create new binding sites that can be recognized by regulatory complexes. And we have sort of like catch-all names for these. Bromodomains are protein complexes that will bind to acetylated regions, and chromodomains are protein complexes that bind to methylated regions in histones. So these modifications can have a ripple, sequential, domino effect. Okay, so we recruit one histone methyltransferase, for example, at one particular site, and it begins to modify in that region and introduce another site of methylation, and that site of methylation might be a site for binding of another chromodomain, and that will recruit more histone methyltransferases to work on neighboring nucleosomes. And you could see that once we begin to do alterations at one site, it can expand to a whole region and then really open up the chromatin to allow expression of a bunch of different genes in that area. One particular example of regulatory proteins that carry out this kind of effect are nuclear hormone receptors. And so I'll spend a little bit of time talking about the action of nuclear hormone receptors and how these can be used to control gene expression. Here are two different hormone receptors. This one, for example, the thyroid hormone receptor, can bind to thyroid hormone, undergoes conformational change that alters its ability to bind with protein complexes. And so when the thyroid hormone is absent, it may bind a protein complex that has histone deacetylase activity, and that might repress expression of genes around this DNA sequence. But when thyroid hormone is present, now the conformational change in that receptor allows for binding of a co-activator protein with histone acetyltransferases, which then modifies the local chromatin structure and activates transcription in this area. In addition to modifications of histone proteins, another really crucial family of chemical modifications is not onto proteins, but is actually onto the DNA itself. And this is the introduction of a methyl group onto cytosine. This is carried out by DNA methyltransferases, and these are used to introduce a mark onto the DNA sequence itself, which also alters its ability to be expressed. Typically, these sites for methylation are present in a palindromic sequence of a CG, so leading 5' to 3'. That CG is also present on the opposite side, CG, so that sequence is present on both strands of the DNA. And these methyltransferases can be thought of to carry out their activity in one of two different ways. Either they can put in a mark where one has not existed before, and we call that de novo methylation. So we're going to move into a palindromic GC sequence, and then we're going to mark the cytosine nucleobase on both strands of the DNA at that site. No methylation present, methylation on both strands. The other type of methylation activity is used to maintain this mark once it's created. We call that maintenance methylation. And the way to think about it is, okay, we have this mark present, the cell divides, we know it's semi-conservative replication, we really understand our Mendelssohn-Stahl experiment, we understand that one strand of this is going to end up in each daughter DNA, so we're going to have one strand with a methylation mark, and the other strand is going to be synthesized without that mark. So if we want to maintain that mark, then we need to recognize that hemimethylated site, and the enzyme will just move in and hit that other complementary strand so that that full mark is maintained from one generation to another. But now this is kind of interesting because it indicates how chemical changes onto the DNA sequence can be used to alter its ability to be expressed and can be passed down from one generation to another generation. This is not a change in the DNA sequence itself, it's a change in a chemical mark of that sequence that can be maintained. So thinking about this issue, a mark does not exist. The cell, for whatever reason, regulatory reason, wants to create it. We call the transcription factors that are responsible for carrying out that activity a pioneer transcription factor. Because these are the transcription factors that will move into these fairly compact DNA regions, recognize a particular DNA site, and then recruit the enzyme that's required to put in that mark, such as cytosine methylation. This is an example of how a transcription factor can be conditionally regulated to be able to introduce those kinds of variations. So this transcription factor, PHO4, is conditionally able to remove nucleosomes, to alter the affinity of the histones for DNA. This is responsive to changes in phosphate concentration. At low phosphate concentration, that transcription factor will not be phosphorylated. It will bind onto the regulatory element in this particular region. It will displace that nucleosome by altering the histone proteins, and then that begins to introduce multiple sites for other regulatory proteins to come in and really open up an expanded area and then allow expression of the mRNA in that region. Some of these sorts of interactions can be really long range, and we talked before about the fact that promoters tend to be really close to the mRNA sequences that they're regulating, but other regulatory regions are very distant. We call those enhancers, and the way that they carry out their effect is by recruiting huge protein complexes, such as the mediator complex, that can associate with transcription factor 2B and create these very large looped architectures for the DNA. It's that kind of organization with this mediator that then brings in RNA polymerase II and then begins the process of transcription in this area. So this mediator plays a key role in coordinating the effects of activators both locally and more distant to a region of genes that need to be regulated. The end result of all of this is very similar to what we were seeing with the lac operon and the trip operon in bacterial systems, which is the recruitment of a polymerase II that can move down the DNA and generate the corresponding mRNA sequence. So here's something that we've given quite a bit of thought to, which is, let's say that the cell is actually not doing a good job of properly regulating gene expression. What opportunities do we have to address this problem and to come up with artificial ways to modulate the activity of aberrant transcription factors and to perhaps therapeutically downregulate the expression of genes associated with different disease states? So there are a whole lot of examples of different transcription factors that can malfunction. They can drive gene expression in cases where they're really establishing a disease state. And many of these, as you can imagine, are associated with cancers. The two that we think about a lot are the androgen receptor as a transcription factor driving prostate cancer. I mentioned that in a different context previously. And estrogen receptor driving early stages of breast cancer. But many of these at this point you will have heard of in the class previously, such as JUN, NF-kappa B, P53. All of these are transcription factors that if they are driving gene expression under conditions where they're not needed, they can be responsible for promoting the growth of cancer tumors. So let's take a little bit more of a look at exactly what happens when one of these transcription factors, such as a steroid hormone receptor, is responding to a signal and driving undesired cellular growth. In the case of the estrogen receptor, this is going to be responsive to the presence of estrogen as a ligand. The binding of the estrogen will induce a change in an alpha helix in that receptor. We'll take a look at that in a second. And that will recruit these co-activator complexes that we've been talking about. For example, these large co-activator assemblies. Here are a family of different steroid hormones, each of which engage with different types of steroid hormone receptor. These steroids, for reasons we've talked about before, are capable of passing through the cell membrane, and once they end up in the cytoplasm, they can engage with the corresponding hormone receptors, allow them to be translocated to the nucleus, where they dimerize and act as transcription factors to turn on responsive genes. Some of these steroid hormones include cortisol or retinoic acid, in addition to testosterone or estrogen. The nuclear hormone receptors have a common architecture. They have a portion of the protein structure that binds to the ligand, the steroid hormone, and they have another portion of the protein structure that has got these DNA binding domains. And you can see here that it looks as though we have a very similar loop turn, helix turn helix type of arrangement in which a helical element is engaging with the DNA regulatory sequence in the major groove. So binding of the ligand then drives this type of activation and association of the nuclear receptor with the DNA. And so this is another picture of what's going on here. The hormone comes in, binds to the nuclear receptor, dimerizes, transits into the nucleus through the nuclear pore, binds on to a DNA response element, and then brings in co-activator proteins to generate the mRNA associated with these target genes. So how can we overcome this type of gene expression? Let's say we're dealing with a cancer that's driven by the action of the estrogen receptor, like prostate cancer. what we need to be able to do is to influence the organization of that estrogen receptor so that it's no longer capable of turning on the genes that are downstream of its regulatory element. This is typically what happens. Estrogen is binding, coactivators associate, genes are activated. And what we need to do is find another ligand that can complex with the estrogen receptor, but can antagonize its activity and prevent coactivators from binding. And this is exactly what the breast cancer therapeutic tamoxifen does. Here's estradiol bound into the estrogen receptor. Here's a helix that plays a crucial role in coactivator binding. And when this therapeutic is bound by the estrogen receptor, it alters the position of this helix so that it no longer is capable of binding its coactivator. Here's two representations of that particular structure right here, present with the estrogen as an agonist or with an antagonist such as tamoxifen. You can see that this helix 12 has been pushed into a new position so that coactivators cannot bind and gene expression is reduced. So our take on this is to take steroid hormones, attach them onto a polymeric backbone so that we can bind into the nuclear hormone receptor, and the presence of this polymer will physically block the coactivator proteins from binding. And we've recently obtained some co-crystal structures of one of the molecules that we've designed in the presence of the estrogen receptor. Here's the steroid hormone that's been conjugated onto one of our polymer molecules. And this shows the way it's bound into the ligand binding domain of the estrogen receptor. Turns out that it is organized in a very unusual way in comparison with most ligands that would activate the estrogen receptor. This shows the way that it is sitting in the ligand binding domain. And here is the mouth of the ligand binding domain. And the presence of that estradiol in that arrangement alters the conformation of this helix 12 in some of the same ways that other anti-estrogens do. And so we've been able to use that type of structure, this covalent attachment of these ligands to these hormone receptors, to alter the ability of coactivator proteins to bind and for these transcription factors to drive the expression of genes downstream of the response elements. We've actually been able to dose these molecules into mice and control the growth of prostate cancer tumors. This shows the way that we've dosed mice that have been implanted with human prostate cancer tumors over the course of 4 weeks. And over that period of time, in the absence of a drug compound, the tumors grow to very large sizes over the course of 3 to 4 weeks. And in the presence of one of these steroid hormone conjugates, we're able to control the growth of those tumors very nicely, precisely because we've been altering the expression of those downstream genes. So that's our take on monitoring and on modulating transcription factors and controlling aberrant gene expression. Did you have a question?
Is there just targets and disease estrogen receptors in therapeutic interventions? And the second part of the question is there a malfunction in the post-translational modification of the actual receptor, or is it something wrong with the DNA that codes for that?
In some cases, there's mutations or alterations of the gene encoding for the estrogen receptor so that it's no longer properly responsive to the ligands. And in other cases, it seems as though there's other influences within the cell that make the regulatory element hypersensitive, or the growth of the cell hypersensitive to the expression of those particular genes. So there's a whole family of different kinds of alterations that can drive either breast cancer or prostate cancer. And you're right that there's some forms of these receptors that have become so mutated that they're no longer responsive to some of these standard anti-estrogens or anti-androgens. And so that really drives the need for new families of drug compounds that can be tailored for those altered steroid hormone receptors. Any other questions about this? Okay, with that, I'm going to get into the subject of epigenetics. So this is a term that we hear a lot about. Does anybody want to take a stab at defining what epigenetics is? Yeah.
What like environmental factors apart from like your genetic sequence that impact your gene expression?
Okay. Anybody else want to add to that definition? Environmental factors that alter gene expression. What's the molecular interpretation of that? How does that manifest itself at the molecular level? So there's some kind of environmental influence that creates a change. What are those changes at the molecular level?
What proteins are produced and in what quantities?
That's probably a downstream effect, but epigenetic modifications, sometimes we call them marks, are the result. Yeah,
Are they like methylations and acetylations in the DNA? and the
exactly.
chromatin?
So we talked about these post-translational modifications, the fact that histones can be acetylated or methylated, and cytosines can also undergo chemical modifications. And I mentioned to you briefly that these alterations can be inherited from one generation to the next. So this is a way to introduce chemical marks onto the DNA sequence. This is one definition of a superimposition on the DNA of a layer of heritable information in the form of chemical changes to either the DNA or the protein within the cell. So these can be introduced as methylation on cytosine bases, and by altering the chromatin structure and the accessibility of the DNA, those influence how the genome is read in different developmental stages, tissue types, or disease states. So it's not a change in the genome sequence itself, the way that we might think about a mutation or a translocation of a chromosomal element, but it's an alteration to the chemical marks that modify how that sequence is read. And I've already introduced you to what those chemical marks are like. Post-translational modifications to proteins, regulatory proteins, and histones, and alterations onto cytosine nucleobases. So this represents the way that these different epigenetic states can be inherited. So these can either be in the form of activator proteins or changes into the DNA itself, and these can be maintained from one generation to another as cells divide. At some point, the epigenetic mark can be established. Again, this can be a change in a histone protein, a methylation mark on cytosine, and then we talked about the way that the cell maintains these marks over time. The same type of epigenetic marks can either be activating or repressive. And this gives two examples of a region of a regulatory component of a DNA sequence. You can see here's the DNA wrapped around the nucleosomes, either in a highly condensed state or in a more open state. So this would correspond to a repressed state, and this would correspond to an activated state. And you can see that in this case, this lysine is unmodified, but this lysine 27 is highly methylated, and this cytosine right here has been methylated. So in this case, these marks indicate a repressed state. But now if we introduce the methylation onto this lysine and demethylate lysine 27, now we have expanded the chromatin, and we're allowing an activated state for this region of the DNA. there's a lot of different types of marks that can be introduced. Methylation, acetylation, and phosphorylation can all be introduced in varying positions on these histone proteins. And this indicates the density with which those post-translational modifications can be made on different positions within a particular histone H3 protein. The only mark that's made on the DNA itself is this mark of methylation on 5-methylcytosine. So there's this really fascinating debate that's going on right now about whether or not epigenetic changes can be manifest as a result of, let's say, a traumatic event, and then those would end up being inherited from one generation to another in the form of epigenetic marks. I find this really fascinating. In some cases, careful studies have been done and seem to indicate that this is in fact the case. So one example is a study of the hunger winter in 1945, when 20,000 people died of starvation in the Netherlands. And the claim is that 70 years ago, when we take a look at the metabolic activity and at the DNA sequences of children who were born during this period of time, we see the presence of these epigenetic marks. For example, the presence of methyl groups in cytosine that altered metabolic activity and have led to a 10% increase in mortality of the children who were born and who were exposed to this type of starvation in utero. So this is very controversial, the very idea that these sorts of epigenetic changes could be evident 70 years later and could correspond to some difference in metabolic activity or maybe even into psychosocial alterations in people's behavior decades later. But in this case, there's a pretty solid theory that methylation marks, epigenetic marks around this PIM3 gene are making it less expressed, and that we're seeing the influence of that many, many years later. So this was from a report that came out at the beginning of this year, and just yesterday, a story came out in the New York Times that's really calling into question the solidity of the science that's being done with studying these epigenetic outcomes of traumatic events. In this case, there was a report that children of prisoners who had been tortured during the Civil War were 10% more likely to die, again, decades later after they had been exposed, after the parents had been exposed to some kind of traumatic event. And I'll just indicate to you that there's a great deal of controversy that these claims are being put forward on, let's say, some flimsy evidence that there is some kind of argument that's being made for transmission of epigenetic information, but that the scientific evidence isn't quite there. And we have a strong emotional appeal because we feel as though images of famine, war, slavery, maybe we recognize the way that it might reverberate generations later. But the general feeling right now is that the epigenetic evidence falls short of demonstrating that past human cruelties affect our physiology today in a predictable or consistent way. This is a very compelling story. Some of you may have heard about these issues. Another one was whether or not there's epigenetic marks in the children of people who were exposed to cruelty in the Holocaust. But the epigenetic data is very flimsy with regard to those studies as well. Sometimes we hear things, we want to believe them, and I'll just encourage you to think about whether or not the data are really good. Overall, think about the operation of the lac operon, how it works, the differences between prokaryotic and eukaryotic gene regulation, how steroid hormone receptors work, and understand the chemical changes that give rise to genetic function. The next lecture will be our last. I'll review what will be on the final exam. I'll talk about siRNA and new genetic therapies, and we'll also talk about the CRISPR-Cas.
Oh my gosh.
I did a lot of
Stop it.
I did it.